Hello everyone. Today we are going to start a new topic, genetics. It is an important topic for CSIR NET exam and it is also an interesting topic. We all know that living organisms reproduce and produce their offspring of same kind. But these offspring do not fully resemble their parents. There is difference in several characters. Individuals of the same species also differ in several characters among themselves. This difference is known as variation. Mechanism by which characters, either resemblances or differences, are transmitted from one generation to the other, that is from the parental generation to the offspring, is known as heredity. And the scientific study of this heredity and variation is known as genetics. So, genetics is the scientific study of heredity and variation. And the term genetics was suggested by William Bateson. Gregor John Mendel is known as the father of genetics. He was an Austrian monk and he performed hybridization experiment on plant and then published his findings in journal and also postulated the principles of inheritance known as the Mendel's law. But scientists at that time ignored his findings. Later on, after his death in 1900, his work was independently rediscovered by three scientists, Hugo de Ries, Karl Correns and Erich Texemark. According to Mendel, traits are inherited or transferred from one generation to other as discrete factors. These factors were termed genes by Wilhelm Janssen. Genes are the unit of heredity. Let us discuss about some of the important terms of genetics. First is allele. We know that factors that control any characteristic in an organism is known as genes. Alternative form of gene that are present on same locus is known as alleles, or in other words, genes that occupy the corresponding position in the homologous chromosome is known as alleles. These control the same characteristics, but their effect may be different. Wild type allele. Allele that are widespread in the population are known as the wild type allele. Proteins that they encode are in the right amount and they function normally, or they have the normal function. But if these wild type alleles are altered by the mutation, then these are known as the mutant allele. And these are present in less than 1% of the population. These encode the proteins whose function and amount is reduced. And these are inherited in the recessive fashion. Dominant allele. Alleles that mask the expression of recessive allele is known as dominant allele, and these are represented by the uppercase or the capital letter. Alleles whose expression is masked by the dominant allele and those express themselves only in the homozygous state is known as recessive allele, and these are represented by the lowercase. All the diploid organisms has two alleles for a trait, and these organisms, diploid organism, may be homozygous or heterozygous. If there are two identical alleles for a trait, then it is known as homozygous. This homozygous may be homozygous dominant. If both the alleles for a trait is dominant, then it is known as homozygous dominant. Homozygous recessive, if both the alleles for a trait are recessive, then it is known as homozygous recessive. Heterozygous, if one of the allele is dominant and the other is recessive, then it is known as heterozygous. Genotype. Genotype is genetic constitution of an individual for any particular trait, and it is represented by symbols depending upon the alleles. Phenotype. It is physical appearance of an individual for any particular trait, and this phenotype depends upon the genotype or genetic constitution of an individual. So Mendel selected garden pea, scientific name Pisum sativum, for its hybridization experiment. It has following advantages. It has well-defined distinct characters. It has bisexual flowers. It mainly self-fertilizes. It was easy to hybridize. It was easy to cultivate, and life cycle was also short. In his experiments on pea plant, Mendel studied 7 characters or traits, and each has 2 variants, dominant and recessive. These 7 characters are stem height, tall, dwarf; flower position, axial, terminal; flower color, violet, white; pod shape, inflated, constricted; pod color, green, yellow; seed color, yellow, green; seed form, round, wrinkled. On the basis of the hybridization experiments on pea plant, Mendel proposed the principles of inheritance, and these are known as the Mendel's law. First is the law of segregation. Law of the segregation was formulated by the monohybrid cross. Cross in which only one trait or character is taken into consideration, and it is applied on the diploid organism. In the diploid organism, for each trait, pair of alleles are present on the same locus of the homologous chromosome. During the gamete formation, these pair of alleles separate, and now each gamete has only one allele for a particular trait. So the gametes are haploid. Gametes are also pure because though the 2 alleles are present together, but these are not influenced by each other, or these do not blend, and when they separate in the gamete, these alleles are discrete or distinct for any trait. This is known as law of purity of gametes. So let us understand this law of segregation by the example of the monohybrid cross performed by Mendel. Parental generation was tall and dwarf. These were purebred. It was homozygous for tall and homozygous for dwarf. So these were crossed. And when these were crossed, gametes were produced. During the formation of the gametes, the pair of allele for each character segregated. That means the homozygous alleles for the tall plant, capital D, segregated and produced the gamete, that is haploid. Similarly, this pair of allele for the dwarf plant separated or segregated and produces the haploid gametes. And F1 generation was all tall plant. But if we see the genetic constitution, it is heterozygous. That means one allele is present for the tall plant, one allele is present for the dwarfness. But when these are present together, only the allele for the tallness is expressed, but the allele for the dwarfness, small t, is concealed. It means allele for the dwarfness of capital T is the dominant allele, and allele for the dwarfness or the small t is the recessive allele. Then this F1 generation was self-fertilized or self-pollinated. F2 generation is produced. This can be represented by the Punnett square. Female gametes are written at the top and male gametes are written at the left side of the square. Here also, during the gamete formation, these 2 alleles segregate. Now we get 3 tall plant and 1 dwarf plant. This is phenotypic ratio. If we see the genetic constitution of these plants, then one is homozygous tall, 2 are heterozygous tall, and 1 is homozygous dwarf. This is genotypic ratio. So from this genotypic and phenotypic ratio, we can conclude that allele for the tallness or the capital T allele is expressed both in the homozygous state and in the heterozygous state. So we get 3 tall plants. Here in the heterozygous plant, both the allele for the tallness and dwarfness is present, but only the allele of the tallness is expressed, or capital T is expressed. That means this capital T allele, that is the allele for the tallness, is dominant, and in the presence of this, the allele for the dwarfness small t is not expressed. So it is recessive, it is masked. But when the allele for the dwarfness is present in the homozygous state, then it is expressed. So small t is the recessive allele, which is only expressed in the homozygous recessive state in the absence of the capital T. Also in the F1 plant, these two alleles, capital T and small t, are present together. In the heterozygous pair, each allele is distinct or discrete for their character. That means these are not influenced by the presence of the other allele, or these do not blend. So the gametes that is obtained, these are pure. This is known as law of purity of gametes. This is why when this recessive allele, though it is present together with the dominant allele in the heterozygous state, when these are separate, these are able to produce the dwarf plant in its homozygous state. This is known as the law of purity of gametes. Now let us see test cross. It is the cross between individual with dominant phenotype and individual with recessive phenotype to determine whether the individual with the dominant phenotype is homozygous or heterozygous. Let us understand this with two scenarios. In the scenario 1, the parental generation tall plant is crossed with the dwarf plant. This is the parental plant. And after the crossing, which we get F1 generation, which is all tall. So what is the genotype of this tall plant? We know that dwarfness is recessive and tallness is dominant. So obviously this dwarf plant is homozygous for the recessive allele, that is small t. So the gamete produced by this dwarf plant is small t. So the tall plant is only possible when this gamete containing the recessive allele fuses with the gamete containing the dominant allele. So all the tall plants will be capital T and small t, that is heterozygous. Now from this we can take out the inference or the conclusion that this tall plant is producing the gamete which consists of the dominant allele. All the gametes produced by this plant has the dominant allele, and when these gametes fuse, then we get all the tall plant which is heterozygous. This means if all the gametes produced by this tall plant has the dominant allele, then the genotype of this tall plant is homozygous. So this tall plant, which is the phenotype of this plant, has the genotype homozygous dominant. Now let us see the second scenario. In this again, the parental generation tall and dwarf is crossed. Here also this dwarf plant is homozygous recessive. Cross is done and we get tall plant, and in the F1 generation we get tall plant and dwarf plant both in the ratio 1:1. So now what is the genotype of this tall plant? From this we can take out that obviously this plant is homozygous recessive. This dwarf plant produces only one type of gamete, which has the recessive allele. So in order to get this dwarf plant, this gamete has to fuse with the gamete produced by the tall plant, which also has the recessive allele. But this is tall plant, so it is also producing gamete which has the dominant allele. That means this tall plant produces two types of the gamete, one carrying the dominant allele and the other carrying the recessive allele. This is possible only if this tall plant is heterozygous. That means its genotype is heterozygous. It is containing both one dominant allele and one recessive allele. So it is producing both the types of the allele, and when the random fusion takes place, then we get both the tall and the dwarf plant in the ratio 1:1. So from this 1:1 ratio of the F1 generation, we can conclude that this tall plant is heterozygous. So from the results that we get due to the test cross, we can always determine whether in the parental generation the plant with the dominant phenotype is homozygous or heterozygous. Law of independent assortment. This was formulated by the Mendel's double hybrid cross, in which two traits are involved or two traits are taken into consideration. Assortment of a pair of alleles for any character is independent of the pair of alleles for any other character during the formation of the gametes. It means a character is independent of any other contrasting character and these are not associated. So when the offsprings are formed, in addition to the parental combination, new combinations are also formed. We can understand this with the example of the dihybrid cross. This is the parental generation. Yellow round seed plant is crossed with the green wrinkle seed plant. Here yellow color is dominant over green color and round seed shape is dominant over the wrinkle seed shape. So the parents are homozygous dominant and homozygous recessive. So they produces one type of the gamete during the gametogenesis, because it is homozygous dominant and homozygous recessive. So the fusion takes place and F1 generation that we get are all yellow and round. It is heterozygous for both the character, that is the genotype is double heterozygous. Then self-fertilization takes place, or it is this F1 generation is self-fertilized. All the four types of the gametes are formed, both male gametes and female gametes. then the F2 generation is produced. This can be represented by the Punnett square. Here these are female gametes and male gamete. Then they fuse randomly, and here 9 are yellow round, 1, 2, 3, 4, 5, 6, 7, 8, 9. 3 are yellow wrinkled, 1, 2, 3. 3 are green and round, 1, 2, 3, and 1 is green wrinkled. So here we see that earlier only 2 types of the combination was present in the parental generation, yellow round seed and green wrinkled seeds. But in the F2 generation, in addition to the parental type, we get the new combinations. How it is possible? It is possible only if the alleles for the seed color assort independently of the alleles for the seed shape, and allele for one character is not associated with the allele for the other character. In the parental generation, we see that the yellow seed color is with round seed shape and the green is with wrinkled. But these are not associated with each other. During the gametogenesis, when F1 is self-fertilized, these assort independently, and in addition to the parental combination, new combination is also formed. That means alleles for the seed color is independent of the alleles for the seed shape. These are not associated. This is the Mendel's dihybrid ratio. This is the phenotypic ratio. So, law of independent assortment has opened various possibilities. It has various advantages. Man can combine all the desired and advantageous characters in commercial crops and farm animals. One can obtain new combination of desired characters and remove the or eliminate the undesired characters. So, it is a boon to agriculture, animal husbandry, horticulture and other fields. So, this is all for Mendel's law of inheritance. In the next video, we will discuss about deviation from these laws of inheritance. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.